So my name's Owen. I'm from the University of Florida, in Florida in the United States. And I work with Dr. Dada on the right. And I'm a third-year PhD student going to be entering my fourth year. And Victor Agboli is a new recruit. He is a first-year PhD student. And we decided to take there's the overview, it's going to be a little longer. We decided to take a little bit of a simpler approach to this, very akin to the keynote this morning. We wanted to set sort of a baseline of what would you do if you took a naive third-year PhD student and stuck them on a difficult challenge of a massive data set with a lot of difficulties. So I'll go over the data set in a little bit more detail. What we were given was a large Excel sheet with a bunch of SRA codes that linked to the NCBI SRA database. They were Ascension codes. And each of these codes linked to a download of FASTQ files. And some of these were from human subjects taken from wounds, some of them from surfaces, some of them from Petri dishes, etc. That can all be found in the descriptions of those websites. Also, there were included things like milliliter solutions of how much antibiotic was added. You get the idea. This was went over in the last talks. One of the things that wasn't discussed, though, is that a lot of these sequences were recorded in different ways. Some of them were single-end, some of them were parent, some of them were long and short reads, and some of them used different sequencing machines. So it created a little bit of difficulty when running a formal bioinformatics pipeline. So we decided we were going to make our own. And once again, this is just a naive solution. So we chose common tools, ResFinder for identifying strains and RefSeq to identify common AMR classes. There was no consistent database for all of these bacteria that we could find readily available. So we had to make one. And so we downloaded all the reference genomes on NCBI from 1990 to 2020, all of them, and we put them together. And we ended up with two references. We ended up with a operational taxonomical unit reference for identifying individual strains and another one for common antimicrobial resistant gene classes. We used these to classify the samples we were given. As far as the samples we were given, we used a really basic approach when figuring out how to process these. We simply loaded the web page using shell, scraped the HTML code entirely, and read the tables and figured out what machine was used, what kind of ends were used, how long the reads were. And using that information, we conducted quality control, trimmed them down, removed dirty data, and repaired them all using the commonly known tools up on the screen now. FastQC, Need Data, Trimmatic, Bowtie, and KMA. Since I have extra time, I don't know if anybody knows anybody that works for these people, but these are phenomenal tools, and this project could not have been done without them. Those sequences, I should go back, after everything was cleaned and repaired, all the samples, we combined them using the two databases there. The strains were taken kind of as an abundance, right? So you're given a sample and it's a long list of genes that's within it. And whenever one of those genes would be referenced or appear within a strain reference, it would kind of get a hit for how many genes were in there. So we were able to get an idea of, so this sample is like 60% from this strain, 20% from this strain, that kind of idea, some prevalence. And we did that for both the O2 and the AMR, and we came up with two input matrices. For each of these matrices, we did the basic thing of dropping features that had no samples and dropping samples with extremely low reads, basically extremely dirty data, which there were some in the training data. For the testing data, we didn't drop anything, even if it was really dirty. And then we took both matrices and normalized them and smacked them together. After we put them together, well, I should go over the isolate metadata. So in those Excel files that had all those Ascension codes, they also had a bunch of other data like collection date, country of source, isolation source, different measurements for how the samples were tested against antibiotics. And we decided we couldn't really use too much of them at all. We attempted to multiple times, and in our training, it would just hurt the model. And honestly, I think it's due to the last bullet point there, the inconsistent reporting. So for instance, in the training data set, we may have a sample with the isolation source labeled as trachea, whereas in the testing data, we may have an isolation source as upper trachea taken at 3 p.m. on a Tuesday after he had brunch. So this kind of inconsistent reporting definitely hurt the ability to correlate the two samples together. I thought of using AI, just throw it into ChatGPT, as simple as that would be, but I wanted to keep it as baseline as possible, and so we just removed the data. In that same way, we removed all of the intermediate. Instead of assuming the intermediate would be relative to susceptible or resistant, in hindsight, I think it would have been a better idea to look at the milliliter solution information and use that as a way to adjust the intermediate. For instance, if you had a really, really small amount of bacteria and a huge amount of antibiotic put in, and it was intermediate, then that could just be really, really resistant, and vice versa. So model training. We used both of these matrices, the O2 and the AMR gene classes, for strain and gene classes, and we set them up with three basic models: one random forest, one logistic regression with lasso, and one XG boost. And we just ran these straight up as binary classifiers using the references that we had, or the features that we had, those being the AMR classes and the strains. And when we ran all three of these models, in the data lab, typically we enjoy ensembles. We find ensembles to be very, very powerful. So we just took all of these three and smacked them together. And because you can't necessarily judge variable importance immediately with an ensemble, we used a very simple leave one out. So we would remove a feature, run the ensemble again, and calculate the accuracy on the training after we did a split. We repeated this twice for stability, and we were able to get some variable importance that I'll show you later. So as far as the model evaluation, the way that we did internal testing, which is this table you're seeing here, is we just took a baseline 70-30 split of the data, and we treated that 30% as test, and then we ran the entire model on the 70%, doing the cross-validation to get the variable get the, I guess, we fine-tuned it. Basically, I don't know the exact wording, but we fine-tuned the data on 70%, and we ran it on 30% to train. And the main reason why we use the ensemble is actually seen on screen right now with this table. All the blue boxes indicate the worst accuracy for the row. And as you can see, the logistic regression with lasso is the first column, the random forest is the second, the XG boost is the third. Each of them have one sample or one bacteria antibiotic combination that they perform the worst in. The ensemble has none of those. In fact, the ensemble performs above the average accuracy of everything except for the second to last row, where it is 0.1 below the average. But that's why we like ensembles. They typically never perform the worst, and that's what you want to do. You want to raise the floor, not fly at the ceiling for one of them. So that's what we did. And our internal record was 86%, and our submission was 84%, and we were tied within a bunch of other people for second place. As for variable importance, using the AMR gene classes, we found pretty basic stuff, right? Things that are typical in most papers, typical in a lot of pharmaceutical research, where tetracycline is in row 2. You can see tetracycline for the drug tetracycline is pretty good at predicting whether or not it's resistant. But we did find some interesting things when it came to the strains, something that I haven't seen other people do yet. But for instance, for a Bumani, hopefully I pronounced that right, LAC4 is actually a really hyper-virulent strain that is used in research, specifically in labs to test antibiotics. And so we found that was a really big predictor. Same thing goes with the FQO2. It's actually an extremely susceptible version, and it's used in resistance training for the gonorrhea as well. The only downside to this variable importance that we didn't do, and in hindsight is kind of mediocre, is we forgot to judge whether or not the accuracy went up or down, and we didn't record it. So unfortunately, we didn't do that. Yeah, major mistake. So I apologize for that. But those are the important ones. As far as going forward with this, there wasn't too much. We wanted to set a baseline. There are things that I could go back and do in hindsight. For instance, the MLs to judge whether or not things were intermediate, could be switched to susceptible or resistant. In addition, using some sort of LLM to go through and actually redefine the inconsistent reporting and get it to a point where we can use that data as a feature. We could also go back and look at just the strains as the means for prediction and then adjust the variable importance to tell us whether or not certain strains will be resistant, and that could be really useful. These are all the references, and one other thing I'd like to add is all of this code is completely publicly available. It's all written in Shell if you like reading that stuff, and it's on my GitHub, so you can go download it if you want. And sorry for the not so long presentation. Any questions?
Any questions? Please.
I didn't think there'd be too many. Oh, there's a microphone up there.
Yeah.
Sorry, because you might have covered this, but I would like. You mentioned how you're using the variables importance to select, to look at the most promising candidates. Why not use a variable selection method? As in, was that a dichotomy? Was
So,
that intentional?
Yeah, the reason we did the variable importance the way we did is because we used an ensemble in the end, right? So we can't necessarily look at what features are important to a specific model. We could go in each model individually and say, like, say XGBoost, select 6 variables is the most. Yeah, we could have done that, but that doesn't necessarily respect the ensemble. And then it comes into the question of why did you prefer these variables? Maybe it's due to the XGBoost, and then stability becomes an issue when we select variables in that way, or features in that way, rather.
Thank you.
Okay, then. Thank you.
Okay.
Very nice.